and welcome to the next chapter. It is growth and development. Growth is a characteristic feature of all living organisms. Most multicellular organisms start life as a single cell and gradually grow into complex organisms with many cells. This involves multiplication of cells through the process of cell division. The quantitative permanent increase in size of an organism is referred to as growth. For growth to take place, the following events must take place. Cells of the organism assimilate nutrients and hence increase in mass. Cell division through mitosis occurs, and this leads to increase in the number of cells. Finally, cell expansion that leads to enlargement and increase in the volume and size of the organism. It is therefore possible to measure the aspects of growth using such parameters as mass, volume, length, height, surface area, and others. On the other hand, development is the qualitative aspect of growth, which involves differentiation of cells and formation of tissues in the body of the organism in order for the tissues to be able to perform specialized function. Development is therefore increase in complexity of the organism. Unlike in the case of growth, it is not possible to measure the actual aspects of development quantitatively, and therefore development can be assessed in terms of increase in complexity of the organism, for example, development of leaves, flowers, and roots. A mature human being has millions of cells in the body, and yet he or she started from a single cell, that is a fertilized egg. During sexual reproduction in mammals, an ovum fuses with a sperm to form a zygote. The zygote divides rapidly without increasing in size, first into 2, then 4, then 8, 16, 32, and so on, till it forms a mass of cells that we call morula. This first cell division is called cleavages. The morula develops a hollow part, resulting into a structure known as blastula or blastocyst. Later, the blastocyst cells differentiate into an inner layer called the endoderm and an outer layer called the ectoderm. The two-layered embryo implants into the uterine wall, and by obtaining nutrients from the maternal blood, starts to grow and develop. As the embryo grows and develops, changes occur in the cell sizes and types. Such changes are referred to as growth and development respectively. This process leads to morphological and physiological changes in the developing young organism, resulting in an adult that is more complex and efficient. In the early stages, all cells of the embryo look alike, but as the development process continues, the cells begin to differentiate and become specialized into different tissues to perform different functions. Growth involves the synthesis of new materials and protoplasm. This requires a continuous supply of food, oxygen, water, warmth, and means of removing waste products. In animals, growth takes place all over the body, but the rates differ in the various parts of the body and at different times. In plants, however, growth and cell division mostly take place at the root tip, just behind the root cap, and stem apex. This is referred to as apical growth, which leads to the lengthening of the plant. However, plants do not only grow upwards and downwards, but sideways as well. This growth leads to an increase in width, by the activity of cambium cells. The increase in width, also called the girth, is termed as secondary growth. Growth can be estimated by measuring some aspects of the organism such as height, weight, volume, and the length over a specific period of time. The measurement so obtained, if plotted against time, results into a growth curve. For most organisms, when the measurements are plotted, they give an S-shaped graph called a sigmoid curve, as shown on your screen. This pattern is due to the fact that growth tends to be slow at first and then speeds up and finally slows down as the adult size is reached. A sigmoid curve may therefore be divided into 4 parts. Part A is called the lag phase, which is a slow phase of growth, and this is the initial phase during which little growth occurs. The growth rate is slow due to the fact that the number of cells dividing are few and the cells have not yet adjusted to the surrounding environmental factors. Phase B, the exponential or log phase. This is the second phase during which growth is rapid or proceeds exponentially. During this phase, the rate of growth is at its maximum, and at any point, the rate of growth is proportional to the amount of material or number of cells of the organism already present. The rapid growth is due to the fact that an increase in the number of cells dividing from 2, 4, 8, 16 all the way following a geometric progression. The cells have also adjusted to the new environment. Food and other factors are not limiting, and hence the cells are not competing for resources, and also the rate of cell increase being higher than that of cell death. Stage C is the decelerating phase, and this is the third phase during which time growth becomes limited as a result of the effect of some internal or external factors or the interaction of both. The slow growth rate is due to the fact that most cells are fully differentiated and are therefore not dividing. Few cells are still dividing. There is also shortage of oxygen, nutrients, and space due to high number of cells, accumulation of metabolic waste products which inhibits growth, and also there is limited acquisition of carbon dioxide as in the case of plants. Phase D, which is called the plateau or the stationary phase, and this is the phase which marks the period where overall growth has ceased and the parameters under consideration remain constant. And this is due to the fact that the rate of cell division equals to the rate of cell death, and that nearly all the cells and tissues are fully differentiated, and therefore there is no further increase in the number of cells. The nature of the curve during this phase may vary depending on the nature of parameter, the species, and the internal factors. In some cases, the curve may continue to increase slightly until the organism dies, as is the case of monocotyledonous plants, many invertebrates, fish, and certain reptiles. This indicates positive growth. In some other cases, the curve flattens out, indicating no change in growth, while other growth curves may tail off, indicating a period of negative growth rate. This negative pattern is characteristic of many, including humans, and is a sign of physical senescence associated with increasing age. However, the sigmoid curve does not apply to all organisms, for example arthropods. Growth takes place at intervals. If volume changes are plotted against time, a different curve is obtained, and this is called an intermittent growth curve, as shown on your screen. The intermittent growth in insects is due to the fact that they have an exoskeleton, and hence growth is possible only when it is shed. This shedding process is known as molting or ecdysis. However, cell division continues to take place during the intermolt phase, but the expansion of tissues is limited by the unshed exoskeleton. Growth and development in plants. The main growth and development phase in plants begin with the germination of the mature seed. Seeds are of two kinds depending on the number of cotyledons or the embryo leaves. A typical seed consists of a seed coat enclosing an embryo. The seed coat is the outer covering, which in most seeds is made up of two layers, an outer testa and an inner one called the tegmen. The testa is thick and the tegmen is a transparent membranous tissue. The two layers protect the seed from bacteria, fungi and other organisms which may damage it. There is a scar called the hilum on one part of the seed, and this is the point where the seed had been attached to the seed stalk or the funicle. Near one end of the hilum is a tiny pore, the micropyle. The micropyle allows water and air into the embryo. The embryo is made up of one or two seed leaves or cotyledons, a plumule or embryonic shoot, and a radicle, the embryonic root. The tip of the radicle is opposite to the micropyle. In some seeds, the cotyledons are swollen as they contain stored food for the growing plumule and radicle. Seeds with only one cotyledon are referred to as monocotyledonous, while those with two are referred to as dicotyledonous. This is the major basis of differentiation between the two large classes of plants, the monocotyledoni and dicotyledoni. Dormancy in seeds. The embryo of a dry, fully developed seed usually passes through a period of rest after ripening period. During this time, the seed performs all its life or physiological processes very slowly and uses little food. This is the period of dormancy. Even if all the favorable environmental conditions for germination are provided to the seed during this period of dormancy, the seed will not germinate. And this is due to the fact that the seed embryo may need to undergo further development before germinating. Some seeds can germinate immediately after being shed from the parent plant, like most tropical plants, while others must pass through the dormancy period lasting for weeks, months, or even years before the seed can germinate. Dormancy provides the seed with enough time for dispersal so that they can germinate in a suitable environment. It also enables the seed to survive during adverse environmental conditions without depleting their food reserves. The embryo has time to develop until favorable conditions are available, for example, availability of water. Several factors cause seed dormancy, and they include: the embryo may not yet be fully developed, presence of chemical inhibitors that inhibit germination in seeds, for example abscisic acid, very low concentrations of hormones like gibberellins and enzymes reduces the ability of seeds to germinate. Hard and impermeable seed coats prevent entry of air and water in some seeds like wattle. In some seeds, the absence of certain wavelengths of light make them remain dormant, for example in some lettuce plants. Also, freezing of seeds during winter lowers the enzymatic activities, rendering them dormant. Dormancy in seeds can be broken in several ways. For example, when the seed embryo are mature, then the seed embryos can break dormancy and germinate. Increase in concentration of hormones like cytokinins and gibberellins will stimulate germination. Favorable environmental factors such as water, oxygen, and suitable temperature. Some wavelengths of light trigger the production of hormones like gibberellins, leading to breaking of dormancy. Finally, sacrification, that is weakening of the tester, is needed before some seeds with hard impermeable seed coats can germinate, and this is achieved naturally by saprophytic bacteria and fungi or by passing through the gut of animals. In agriculture, seeds of some plants are weakened by boiling, roasting, and cracking, for example, wattle trees. Germination of seeds. The process by which the seed develops into a seedling is known as germination. It refers to all the changes that take place when a seed becomes a seedling. At the beginning of germination, water is absorbed into the seed through the micropyle in a process known as imbibition and causes the seed to swell. The cells of the cotyledon become turgid and active. They begin to make use of the water to dissolve and break down the food substances stored in the cotyledons. The soluble food is transported to the growing plumule and radicle. The plumule grows into a shoot while the radicle grows into a root. The radicle emerges from the seed through the micropyle, bursting the seed coat as it does so. Seeds need certain conditions to germinate and grow, and some of these conditions are external, for example water, oxygen, and suitable temperature, while others are internal, such as enzymes, hormones, and viability of the seeds themselves. Water. A non-germinating seed contains very little water. Without water, a seed cannot germinate. Water activates the enzymes and provides the medium for enzymes to act and break down the stored food into soluble form. It also hydrolyzes and dissolves the food materials and is also the medium of transport of dissolved food substances through the various cells to the growing region of the radicle and plumule. Besides, water softens the seed coat, which can subsequently burst and facilitate the emergence of the radicle. Oxygen. All germinating seeds require energy for cell division and growth. This energy is obtained from the oxidation of food substances stored in the seed through respiration, and thus making oxygen an important factor in seed germination. Seed in waterlogged soils or seed buried deep into the soil will not germinate due to lack of oxygen. Temperature. Most seeds require suitable temperature before they can germinate. Seeds will not germinate below 0 degrees Celsius or above 47 degrees Celsius. The optimum temperature for seed to germinate is 30 degrees Celsius. At high temperature, the protoplasm is killed and the enzymes in the seed are denatured. At very low temperatures, the enzymes become inactive. Therefore, the protoplasm and the enzymes work best within the optimum temperature range. The rate of germination increases with temperature until it reaches an optimum, and this varies from plant to plant. Enzymes play a vital role during germination in the breakdown and subsequent oxidation of food. Food is stored in seeds in form of carbohydrates, fats, and proteins, which are in insoluble form. The insoluble food is converted into a soluble form by enzymes. Carbohydrates are broken down to glucose by diastase enzymes, fats to fatty acids and glycerol by lipases, and proteins into amino acids by protease. Enzymes are also necessary for conversion of hydrolyzed products to new plant tissues. Hormones play a vital role in plant germination as they act as growth stimulators. Such hormones include gibberellins and cytokinins, and these hormones also counteract the effect of germination inhibitors. Viability. Now, only seeds whose embryo are alive and healthy will be able to germinate and grow. Seeds stored for long periods usually lose their viability due to depletion of food reserves and destruction of their embryo by pests and diseases. The nature of germination varies in different seeds depending on whether the cotyledons remain under the soil or are brought up the surface, giving rise to two types of germination, epigynous and hypogynous. In epigynous germination, the radicle grows out through the micropyle. It grows downwards into the soil as a primary root from which other roots arise. The part of the embryo between the cotyledon and the radicle is called the hypocotyle. This part curves and pushes upwards through the soil, protecting the delicate shoot tip. The hypocotyle then straightens and elongates, carrying with it the two cotyledons, which turn green and leafy. They start manufacturing food for the growing seedling. The plumule, which is lying between the two cotyledons, begin to grow into first foliage leaves, which start manufacturing food. In hypogynous germination, like in maize, the endosperm provides food to the embryo, which begins to grow. The radicle, along with the protective covering, grows out of the seed. The epicotyle is the part of the embryo between the cotyledon and the plumule. The epicotyle elongates and the plumule grows out of the coleoptile and forms the first foliage leaves. The seedling now begins to produce its own food and the endosperm soon shrivels. This type of germination, in which the cotyledon remains below the ground, is known as hypogynous germination. Therefore, hypogynous germination is a type of germination in which the cotyledons remain below the ground. Primary and secondary growth. The region of growth in plants is found in localized areas called the meristems. A meristem is a group of undifferentiated cells in plants which are capable of continuous mitotic cell division. Primary growth occurs at the tip of roots and shoots due to the activity of the apical meristems. These meristems originate from the embryonic tissues. In this growth, there are three distinctive regions: the region of cell division, the region of cell elongation, and the region of cell differentiation. The region of cell division is an area of actively dividing meristematic cells. These cells have thin cell walls, dense cytoplasm, and no vacuoles. The cells become enlarged to their maximum size by the stretching of their cell walls. Vacuoles start forming and enlarging. In the region of cell differentiation, the cells attain their permanent size, have large vacuoles and thickened cell walls. The cells also differentiate into tissues specialized for specific functions. Primary growth results to an increase in the length of shoots and roots. Secondary growth results in an increase in width or girth due to the activity of cambium. In secondary growth, new tissues are formed by the vascular cambium and the cork cambium. In monocotyledonous plants, there are no cambium cells in the vascular bundles. The growth in diameter is due to the enlargement of the primary cells. So, increase in girth in dicots is accomplished by secondary growth involving the vascular cambium, which is a meristematic tissue between the phloem and xylem. In a young plant, vascular bundles composed of xylem, phloem and cambium are distinct from one another. As growth continues, the vascular cambium forms a continuous ring near the perimeter of the stem. As cambium cells divide, differentiating cells on the interior become secondary xylem, and those on the exterior become secondary phloem. Girth increases as secondary xylem accumulates. The old phloem cells are crushed as the cambium moves outwards. Rings are visible on the wood. They are the result of cells forming during the wet and dry seasons and are used to estimate the ages of woody plants. Bark replaces the epidermis on the exterior of the trunk, as the meristem just near the epidermis produces cork cells. We now look at the role of growth hormones in plants. Now, plant hormones are chemicals produced in very small amounts within the plant body, and they play a very important part in regulating plant growth and development. Most growth hormones are produced at the tip of a shoot and transported downwards to the root. The root tip produces very small quantities of the hormones. There are many different types of plant hormones, and one well-known group is the auxins. Indole acetic acid is one best-known auxin. Auxins are produced at the shoot and root tips. Auxins are known to have various effects on the growth and development in plants. They stimulate cell division and cell elongation in stems and roots, leading to primary growth. Auxins cause tropic responses, which are growth responses in plants due to external stimuli acting from a given direction. On the other hand, indole acetic acid stimulates the growth of adventitious roots, which develop from the stem rather than the main root. Cuttings can be encouraged to develop roots with the help of indole acetic acid. If the cut end of the stem is dipped into indole acetic acid, root sprouting is faster. Indole acetic acid is also used to induce parthenocapy, which is the growth of an ovary into a fruit without fertilization. This is commonly used by horticulturalists to bring about a good crop of fruits, particularly pineapples. Auxins are known to inhibit the development of side branches from lateral buds, and therefore they enhance apical dominance. During secondary growth, auxins play an important role by initiating cell division in the cambium and differentiation of these cambium cells into vascular tissues. Auxins, in association with other plant hormones such as cytokinins, induce callus tissue, which causes healing of wounds. When the concentration of auxins fall in the plant, it promotes formation of an abscission layer, leading to leaf fall. A synthetic auxin, 2,4-dichlorophenoxyacetic acid, induces distorted growth and excessive respiration, leading to death of the plant. It is therefore used as a selective weed killer. Gibberellins are another important group of plant hormones. Gibberellins are a mixture of compounds and have a very high effect on growth. The most important in growth is gibberellic acid. Gibberellins are distinguished from auxins by their stimulation of rapid cell division and cell elongation in dwarf varieties of certain plants. Dwarf conditions are thought to be caused by a shortage of gibberellins due to a genetic deficiency. Gibberellins are important in fruit formation. They induce the growth of ovaries into fruits after fertilization. They also induce parthenocapy. Gibberellins also promote the formation of side branches from lateral buds and breaks dormancy in buds. This is common in species of temperate plants whose buds become dormant in winter. In addition, this hormone also inhibits sprouting of adventitious roots from stem cuttings. It retards formation of an abscission layer and hence reduces leaf fall. Gibberellins also break seed dormancy by activating the enzymes involved in the breakdown of food substances during germination. Cytokinins, also known as kinetins, are growth substances which promote growth in plants when they interact with auxins. In the presence of auxins, they stimulate cell division, thereby bringing about growth of roots, leaves, and buds. They also stimulate formation of the callus tissue in plants. The callus tissue is used in the repair of wounds in damaged parts of the plant. Cytokinins promote flowering and breaking of seed dormancy in some plant species. They also promote the formation of adventitious roots from stems and stimulate lateral bud development in shoots. When in high concentration, cytokinins induce cell enlargement of leaves, but in low concentration, they encourage leaf senescence and hence leaf fall. Ethylene is a growth substance produced in plants in gaseous form. Its major effect in plants is that it causes ripening and falling of fruits. This is widely applied in horticultural farms in ripening and harvesting of fruits. It stimulates the formation of an abscission layer leading to leaf fall. Induces thickening of stems by promoting cell division and differentiation at the cambium meristem, but it inhibits stem elongation. Ethylene promotes breaking of seed dormancy in some seeds and flower formation mostly in pineapples. Abscisic acid is a plant hormone whose effects are inhibitory in nature. It inhibits seed germination, leading to seed dormancy. Inhibits sprouting of buds from stems and retards stem elongation. In high concentration, abscisic acid causes closing of the stomata. This effect is important in that it enables plants to reduce water loss. It also promotes leaf and fruit fall. Another hormone, florigen, is produced in plants where it promotes flowering. Growth and development in insects. Now, majority of insects lay eggs that hatch into larvae, which is an immature stage usually quite different from the adult ecology and behavior. Depending on the insect species, the larva is referred to as grub, maggot, or a caterpillar. Generally, the larva eats a lot, grows rapidly, and sheds its cuticle several times until it reaches full size to become a pupa. The pupa is an inactive, non-feeding stage during which extensive breakdown and reorganization of body tissues occur, eventually giving rise to the imago or adult form. Such changes are called metamorphoses and occur in butterflies, moths, bees, wasps, and flies. Insects which pass through these stages, namely egg, larva, pupa, and into adult, in their development are said to undergo complete metamorphosis. An example of complete metamorphosis is the development of a housefly. Now, the eggs of a housefly are laid in batches of between 100 and 150. They measure about 1 millimeter in length. After about 5 hours, the eggs hatch into larvae, which are called maggots. The maggots feed on dead, decaying organic matter. After several molts and increase in size, a larvae reaches about 1 centimeter in length, and this takes about 5 days. After this, the larvae changes into pupa encased in a pupal case called a puparium, from which the adult fly will later emerge. Now, the emergence of the adult fly involves the breaking of the puparium by inflating of the egg, which is elastic at least for a short while, and the adult housefly emerges. After emergence, the adult takes about 2 weeks of feeding and growing to attain sexual maturity. That is, the males can mate and the females are able to lay eggs. Other insects go through only 3 stages in their life cycle before attaining maturity into adulthood. This is known as incomplete metamorphosis and is seen in insects such as grasshoppers and cockroaches. For example, in a grasshopper, the eggs are laid and covered with a layer of soil in the ground. When conditions are favorable, a nymph hatches out of the eggs. Now, a nymph resembles an adult in all aspects except that it is wingless and lacks sexual maturity. The nymph undergoes several molts called instars before attaining maturity. After molting, the nymph increases in size until it is ready to mature into an adult. Now, next is the role of hormones in insect metamorphosis. Insect metamorphosis is controlled by hormones which are produced in 3 glands, namely the neurosecretory cells in the brain ganglia, a pair of corpora alata located in the mandibular segment, and prothoracic glands in the thorax. The neuroendocrine system consists of neurosecretory cells that secrete hormones. Neurosecretory cells are mainly located in the brain, ventral nerve cord, and the corpora cardiaca. They are also found in association with other nervous tissues located throughout the body. Neurohormones are the master regulators and control most physiological activities, including regulating the secretion of the hormones that control molting, metamorphosis, and reproduction. Corpora cardiaca are the main endocrine structures attached to the brain. Neurosecretory cells located in the brain synthesize and transport neurohormones to the corpora cardiaca, from which the hormones are stored and released. In addition, corpora cardiaca contains intrinsic neurosecretory cells that also synthesize and release neurohormones. The corpora alata is associated with the corpora cardiaca, but they are not part of the neuroendocrine system. The corpora alata are associated with the corpora cardiaca, but they are not part of the neuroendocrine system. Corpora alata synthesize and release juvenile hormone. The prothoracic glands are grape-like clusters of cells surrounding the trachea in the first thoracic segment. They secrete ecdysone, the hormone that stimulates molting, a process necessary for insect growth. They disintegrate in adult insect because adult insects no longer molt. During larval stages of the insect, the Corpora alata produces juvenile hormone. This leads to formation of larval cuticle, therefore molting does not go beyond the larval stage. When the larva matures, the corpus alatum disintegrates. At this time, the neurosecretory cells stimulate the prothoracic glands to produce molting hormone, the ecdysone. Ecdysone is responsible for molting in insects, leading to laying of adult cuticle.
Without many of these discoveries and inventions, few of us would be alive today. Welcome to WatchMojo.com, and today we're counting down our picks for the top 10 greatest medical breakthroughs in history. Number 7. Painkillers. In the 1800s, two of the most important analgesics were introduced to the world, morphine and aspirin. Since then, the pain-killing properties of the naturally occurring chemical compound known as morphine have lessened the suffering of an immeasurable number of human beings and have led to the development of many other analgesics. Meanwhile, German chemist Felix Hoffmann's synthesis of acetylsalicylic acid into aspirin for medical purposes has turned the drug into one of the planet's most popular medications for pain relief.